Irene Turnbull. I'm assistant professor at the Icahn School of Medicine at Mount Sinai in New York. We're at BCBS 2023, and we just finished a fantastic session on arrhythmogenic basis of cardiac disease. And I'm here with two of the fantastic presenters that we had today, who will give us a snippet on what was discussed today. Dr. Wu, please.
Yeah, I'm Sean Wu. I'm a professor in cardiology and also the associate director for the Stanford Cardiovascular Institute. I am working on heart development, and I will be talking about understanding the enhancers that regulates the earliest steps of heart formation.
Dr. Alvarado.
And my name is Francisco Alvarado. I'm an assistant professor of cardiovascular medicine at the University of Wisconsin in Madison. And I taught, and my lab studies how, what are the mechanisms that lead to sudden death and cardiac arrhythmia. And I talked in this session about a mutation in one calcium handling protein in the heart, RyR2 receptor 2, which leads to arrhythmogenic cardiomyopathy in a very exciting rabbit model that we generated.
Dr. Wu, can you tell us a little more on this understanding of the comprehensive mapping of the enhancers in the development heart and relevance?
Yeah, sure. So when we started out trying to look at how genes are regulated during development, there has been a long history of different technologies that are available, and particularly in recent years, the use of single-cell profiling has become quite popular, and one of the most recent advances is to be able to combine both the genes that are expressed within the same single cell with the epigenetic profiles, such as the chromatin openings within the same cell, using assays called attack-seq. Mm-hmm. And we decided that in this study to be able to see for all of the developing heart cells what are the correlation between the opening of the chromatin from the attack-seq and also what are the genes that are expressed coincident within the same cell, so that we are able to see how the regulatory activities at the chromatin is reflected in the genes that are being driven up or down during the process of development. And we found quite interesting relationship between certain parts of the enhancer that are able to directly correlate with the onset of gene expression. And while there's large number of enhancers that are always present and do not necessarily specifically have a participatory role in turning on or off gene expressions. So what this work, which largely at this point is a profiling work, now give us a lot of interesting hypotheses to which areas within the regulatory parts of the genome is actually the key responsible region for the expression of certain genes. And that now gives us an opportunity to be able to go forward to start doing specific experimental perturbation to determine the significance of the various different regulatory parts to any one important cardiac gene. So we're super excited by what the new data set that we have done in this work to now be able to give us more insights on how these very important but yet very complicated genes are regulated during heart development.
Absolutely fascinating work, Dr. Wu. Thank you very much. And Dr. Alvarado, speaking on the RyR2 receptor dysfunction in the context of cardiomyopathy, can you tell us a little bit on the relevance and the importance, how it relates with the model that you found and the association with clinical disease?
Sure. So our favorite ion in the lab is calcium. Calcium is essential for heart contraction. And over many years, there is a lot of evidence suggesting that dysfunction of RyR2 receptor, which is a major calcium release channel in the heart, leads to sudden death and arrhythmia, especially in young kids that have structurally normal hearts. There has been a lot of speculation for many years in the field that since RyR2 receptors are calcium channels, that perhaps dysfunction of this protein also leads to structural remodeling of the heart and cardiomyopathy. However, there have never been animal models that recapitulate any of these phenotypes. So we decided to study one mutation that we introduced in a rabbit model. And the reason we chose a rabbit model is because the calcium handling machinery is closer to a human than the mouse. The mouse is the model that we usually use because it's easy to modify genetically. But we chose a rabbit because of the similarity with the human. The big surprise that we got was that this one mutation that only generates arrhythmia in the mouse model also generates arrhythmogenic cardiomyopathy in the rabbit. Arrhythmogenic cardiomyopathy is progressive, and it starts just showing us arrhythmias, but then when the disease progresses, the heart dilates and it eventually fails. So right now what we're trying to understand is how this one mutation can produce a progressive phenotype that covers that broad phenotypic spectrum from an arrhythmia to a cardiomyopathy. And this is very important because understanding how calcium dysfunction leads to heart disease also opens a lot of doorways that we can use to create new therapies that can improve the life of patients that have not only cardiomyopathies like arrhythmogenic cardiomyopathy, but other diseases that also involve calcium mishandling.
Thank you, Dr. Alvarado. That's a fantastic work as well, and very exciting to learn more in the future on the progress of this research, both with the mapping of the enhancers and all the future validation studies, and also the studies that you'll be doing in the rabbit model. And we also had other talks, and overall this was a very interesting session, and we learned on the great progress that's being made in this area and very clinically very relevant area of arrhythmogenesis in cardiac disease.